Hello students, my name is Niyati Said and thanks for watching Edupedia word videos. My topic for the presentation is the fourth section of the chapter Excretion and Osmoregulation, that is excretory products and their elimination. In this presentation we'll be studying about the mechanism of urine formation, that is physiology of excretion. Okay. So, the formation of urine takes place in 3 steps. Which are those? First is ultrafiltration. This is ultrafiltration, and then reabsorption takes place, and then secretion takes place. In ultrafiltration, you can say that it is a physical process of filtration of blood under high pressure that takes place in the Malfigian body. Now, what is Malfigian body? Malfigian body is, you can say that it consists of Bowman's capsule as well as glomerulus. Okay, so here Bowman capsule acts as an ultrafilter. Okay, this is Bowman's capsule which acts as an ultrafilter. The main structure involved are visceral epithelium of Bowman's capsule and glomerulus. Okay. The wall of the glomerulus capillaries, as you can see, these are the glomerulus capillaries. So the wall of the glomerulus capillaries is made up of a single layer of cells which we call it as endothelium. Okay, the wall of the capillaries is fenestrated, that is, it has numbers of pores which allow most of the constituents of blood plasma to pass except plasma proteins and blood cells. Okay, now comes reabsorption. This is the second step of the mechanism of the urine formation. In reabsorption, what happens is the glomerular filtrate formed in the urinary space after ultrafiltration. Okay, after ultrafiltration contains some nitrogenous waste as well as some useful substances such as salt of Na+, K+, Ca2+, Cl-, amino acids, vitamins, glucose, water, etc. So the useful substances need to be reabsorbed in the water. Okay, it needs to be reabsorbed in the water, and that's how selective reabsorption, which is the second step of the mechanism of urine formation, takes place or come into being. That it helps in reabsorbing the useful substances in the blood back. Okay. So this is how a second step takes place, that is selective reabsorption. And then final is tubular secretion. Tubular secretion is the third step in the urine formation. Although the blood is ultra-filtered in Bowman's capsule, some amount of blood escape ultrafiltration. Okay, so this blood circulates in capillaries around the renal tubule and it contains nitrogenous waste. Okay, I repeat that although the blood is ultrafiltered in the Bowman's capsule, as I have just taught you, but it is seen that some amount of blood escapes ultrafiltration. Okay, so this blood circulates in capillaries around the renal tubule and it contains nitrogenous waste. Okay, and from these capillaries the nitrogenous waste like ammonia, creatinine as well as ions such as K+, H+ are absorbed actively and then discharged in the cavity of the renal tubule. So thus you can say that tubular secretion is just opposite to selective reabsorption, which is the second step. Okay, and it occurs, the tubular secretion, it takes place in proximal tubule as well as convoluted tubule. Okay, so this is how I summarized the 3 steps of the formation of the urine. Okay, let's see this diagram. This is Bowman's capsule, which is also you can say Malfigian tubule, because Malfigian tubule consists of Bowman's capsule as well as glomerular capillaries. Okay, these red one are glomerular capillaries. So this helps in the filtration of smaller molecules. Okay, and then comes proximal tubule. In proximal tubule, as I have just told you, that secretion takes place in the proximal tubule. Okay, so the active secretion of weak electrolytes and reabsorption of water takes place in proximal tubule. Okay, and then comes distal tubule. In this, passive transfer of lipid soluble drugs and reabsorption of water takes place. Okay, and then comes the loop of Henle, in which reabsorption of water takes place. This is loop of Henle. Okay, this is loop of Henle, in which reabsorption of water takes place. So this is the nephron of the kidney, which is an structural and functional unit of kidney. It is about 40 to 60 nanometer in length. Okay, structurally it consists of two main parts. First is Malfigian body, or it is also known as Malfigian corpuscle, and other is known as tubular region. Okay, Malfigian body, as I have said, that they can also be called as renal corpuscles or Malfigian corpuscles. Okay, and it consists of Bowman capsule and the glomerular capillaries, which is this. Okay, with the one which is indicated by red in color. Okay, Bowman capsule, this is Bowman capsule. Bowman capsule, it is cup shaped, it is double walled structure, and its outer wall is called parietal epithelium, and it is made up of flat squamous epithelium. Okay, and the inner wall is called as visceral epithelium, and it is made up of specialized cells, which we call it as podocytes. Okay, podocytes, they are modified squamous epithelium cells. They produce foot-like projections, which we call it as pedicles, which extends up to the glomerulus. Okay, and the space enclosed by the parietal and the visceral epithelium in Bowman capsule is which we call it as urinary space. Okay, so this was, I summarized the 3 steps of the formation of urine. Now let's see in detail. What is ultrafiltration? I have told you that mechanism of urine takes place in 3 steps. First is ultrafiltration, second is reabsorption, and third is tubular secretion. Okay, now comes ultrafiltration. Ultrafiltration, as I have told you, that it takes place in the Bowman's capsule. Okay, and the renal artery, which brings blood to the kidney, is split up into numerous arterioles, each feeding a nephron. See, this we call it as glomerular apparatus. Okay, so this is how renal artery, it splits up into the numerous arterioles, each feeding a nephron. Now the arterioles, they split up into numerous capillaries, which form a knot, which we call it as glomerulus. Okay, see, this is glomerulus, the red knot-like structure. This is glomerulus. Okay, and the arterioles, they split up into numerous capillaries, which form a knot-like structure, which we call it as glomerulus. And this is surrounded by Bowman's capsule, as I have shown you in the previous figure, that Bowman's capsule, it surrounds the entire glomerulus, that is the knot of the arterioles. Okay, so the arterioles leading into the glomerulus is wider than one leading out, so there is a high blood pressure in the capillaries of the glomerulus. Now question may come, why capillaries of the glomerulus, they have high blood pressure? Okay, so you can say that the arterioles leading into glomerulus is wider than the one that leads out, and that's the reason that high blood pressure in the capillaries of glomerulus is seen. Okay, so this pressure forces plasma out of the blood by ultrafiltration. See, this is the significance of having a high blood pressure in the arterioles. Because of the high blood pressure, it forces plasma out of the blood by ultrafiltration. Now, once the blood is filtered, in this way, only blood cells and large plasma proteins remain in the blood, and they continue into the PCT, that is proximal convoluted tubule. Okay. I'll show you how it takes place. See, this is afferent arteriole, okay, and this is efferent arterioles. And what is this? This is a glomerulus. That is, you can say that it is a knot of arterioles. Okay, so this is glomerular capillaries, and from here blood enters from afferent arterioles, and because of the high blood pressure, it pushes plasma out of it. Okay, and the afferent arteriole is, you can say that afferent arteriole is a branch of renal artery that enters the Bowman capsule, and it forms a knot of capillaries which we call it as glomerulus capillaries. The blood leaves the glomerulus through efferent arteriole. See, this is efferent arteriole. Okay, and the diameter of the afferent arteriole, see, the diameter of the afferent arteriole is wider than that of the efferent arteriole, and hence the blood in the glomerulus is under pressure. Okay, due to the pressure, the blood that passes through the capillaries of the glomerulus is ultra-filtered, and that is what we are studying. The first step of the mechanism of urine formation, that is ultrafiltration. Because of the high pressure in the glomerulus capillaries, blood passes through the capillaries of the glomerulus is ultra-filtered, and during the ultrafiltration, blood cells cannot pass through the pores, and hence only blood plasma is filtered. Okay, that means the blood devoid of blood cells is known as plasma. Okay, practically all the components of plasma except the larger molecule of plasma protein pass through the pores, and they appear in the cavity of Bowman's capsule. Now, this filtrate which takes place in the ultrafiltration is known as glomerular filtrate. Okay, or it is also known as primary urine. It has both useful materials such as glucose, amino acids, salts, as well as waste materials such as urea from the blood. The filtrate is isotonic or similar to blood plasma. Okay, and thus ultrafiltration is passive and non-selective, and the rate of the ultrafiltration is about 180 liters per day. Okay, now moving on forward to selective reabsorption, which is the second step in the urine formation. Okay, so what happens in selective reabsorption? As we just read that the glomerular filtrate, okay, the ultrafiltration filtrate, it formed in the urinary space after ultrafiltration, contains nitrogenous waste as well as some useful substances such as salt of Na+, K+, Ca2+, and Cl-, water, glucose, vitamins, etc. The useful substances need to be reabsorbed in the blood. So the glomerular filtrate, it passes through the renal tubule. See, this is the second step, and in this what happens is, see, this is ultrafiltration. Ultrafiltration we have already studied, and then comes second is reabsorption. So this is second. Okay, in this what happens is the glomerular filtrate, which we just studied, the ultrafiltration product, it passes through the renal tubule. Okay, this is renal tubule. Okay, So it passes through the renal tubule, where selective reabsorption of useful substances takes place from renal tubule to blood in the peritubular capillaries. This is peritubular capillaries. Okay. So the renal tubule comprise of a proximal convoluted tubule, a loop of Henle, distal convoluted tubule, and terminal duct. Now, the inner lining of the renal tubule is made up of columnar, cuboidal, and ciliated epithelium. The cells of the columnar epithelium, it show microvilli, which increases the surface area for reabsorption. The cells also have many mitochondria to provide energy for active absorption. Various regions of the tubules are specialized for various activities. Okay. So this is how selective reabsorption takes place, and you will be amazed to know that selective reabsorption occurs in the PCT, that is proximal convoluted tubule. This is the longest and the widest part of the nephron. What is nephron? Nephron is a functional and structural unit of kidney. In this part, over 80% of the filtrate is reabsorbed into the blood, ensuring all useful material is returned to the blood. Okay, all glucose, all amino acids, and 85% of the mineral ions are reabsorbed by active transport. Small proteins are also reabsorbed. 80% of the water is absorbed back into the blood by osmosis. Okay. And the proximal convoluted tubule cells, they have mitochondria that provides ATP for active transport, and microvilli to increase the surface area for absorption. Okay, see, this is how it takes place. In selective reabsorption, see, sodium, amino acids, and glucose, they are reabsorbed in the filtrate, reabsorbed from the filtrate back into the blood. Okay, blood flows in the capillary. Okay, so it is filtrate back into the blood, and this is how selective reabsorption takes place. Now, please note that ascending limb of loop of Henle is permeable to electrolytes and impermeable to water. As the filtrate passes up the ascending limb, electrolytes, they are reabsorbed actively or passively into medullary fluid. Okay, so this is how selective reabsorption takes place, in which useful substances such as Na+, K+, Cl-, Ca2+, amino acids, glucose, vitamins, water, they all are reabsorbed in the blood. Okay. Now moving on to loop of Henle, as I have told you that the loop of Henle, it creates the condition for final reabsorption of water in the collecting duct by creating an ion gradient. Okay, I've told you that the tubular fluid, it passes down the descending limb of the loop of Henle. The descending limb is permeable to water but almost impermeable to electrolytes. Water is progressively reabsorbed when filtrate passes down the descending limb, and hence the filtrate becomes progressively hypertonic. Okay. And in the loop of Henle, salt is added to the filtrate in the descending limb. The cells surrounding are impermeable to ions, I have told you, and due to water moving from a less negative to a more negative water potential, water comes into the filtrate. And in the ascending limb of loop of Henle, the walls are impermeable to water, so the water remains and the ion leave, creating an ion gradient in the medulla. Now moving on to the third process of the mechanism of urine formation, that is tubular secretion. Tubular secretion is also known as renal secretion, and it is the third step in the urine formation mechanism. Although the blood is ultrafiltered in Bowman's capsule, some amount of blood escapes ultrafiltration, okay, and this blood circulates in capillaries around the renal tubule, and it contains nitrogenous waste. From these capillaries, the nitrogenous waste like ammonia and creatinine, as well as ions such as K+, H+, they are absorbed actively and then discharged in the cavity of the renal tubule. Thus, this process is opposite to selective reabsorption, which is the second step in the urine formation. And this step, the tubular secretion step, it occurs in proximal and distal convoluted tubule, that is PCT and DCT. Okay. The secretion of hippuric acid, creatinine, ammonia, and certain drugs, they take place in PCT. PCT also helps to maintain pH and ionic composition of body fluids by selective secretion of hydrogen ions, ammonia, potassium ions into the filtrate, and by absorption of HCO3- from it. Okay, so this is how the third step takes place, and tubular secretion occurs from the epithelial cells that line the renal tubules and collecting duct. Okay. See, this is how tubular secretion takes place, in which, as I have explained you, that although the blood is ultrafiltered in Bowman's capsule, some amount of blood escapes ultrafiltration. So this blood circulates in capillaries. As you can see, this blood circulates in the capillaries, and capillaries around the renal tubule. See, this is the renal tubule, and this is the blood capillaries, and this blood, it circulates in the capillaries around the renal tubule, and it contains nitrogenous waste such as urea, drug, etc. So from these capillaries, the nitrogenous waste like ammonia, creatinine, urea, and ions such as K+ and H+ are absorbed by this renal tubule, that is distal tubule or collecting tubule. So they are absorbed actively and then discharged in the cavity of the renal tubule. Okay, this is the cavity of the renal tubule. Thus, this process is opposite to selective reabsorption. Okay, and why is it different or opposite from the selective reabsorption step? Because remember, students, that in selective reabsorption, the glomerular filtrate formed in the urinary space after ultrafiltration contains nitrogenous waste as well as some useful substances such as Na+, K+, amino acids, glucose, vitamins, water, and the useful substances, they need to be reabsorbed in the water. So in that selective reabsorption of useful substances took place from renal tubule to blood in the peritubular capillaries. Okay, but here it is just opposite. Okay. So and remember, students, that the secretion taking place in the renal tubule is not real secretion, and the renal cells, that is renal cells, they do not synthesize any material and add it to urine, but they only allow certain substances from blood to pass into the tubule or cavity of the tubule, okay, or lumen of the tubule. Now, what is the role of collecting duct in tubular secretion step? This long duct, okay, this is the long duct which extends from the cortex of the kidney to the inner part of the medulla. See, kidney has two parts, cortex and medulla. So this long duct the significance of the collecting duct in tubular secretion step or the mechanism of urine formation is that this long duct, it extends from the cortex of the kidney to the inner parts of the medulla. Large parts of water could be reabsorbed from this region to produce a concentrated urine. And this steps allows passage of a small amount of urea into the medullary interstitial fluid to keep osmotic concentration. It also plays role in maintenance of pH and ionic balance of blood by selective secretion of H+ and K+ ion. Okay, so this was all about the mechanism of the urine formation. In my next section of the presentation, we'll be studying about the composition of urine and the factor that influences the urine formation. Okay, so till then, stay tuned and keep watching Edupedia Word with